Hey, howdy, hey folks, it is Mrs. Johnson with the Active Wisdom channel. We are doing color genetics once again. We're going to do some simpler concept practice before we get into true color genetics of like livestock and agriculture and all the things. Because some of these simpler concepts are easier to practice on less convoluted things before we jump into the crazy cool madness that is color genetics in livestock. So, let's do that. First and foremost, concept practice. Pause the video after my spiel if you'd like to practice. But what vocab words do you remember? What concepts do you remember? Try and list as many as you can. Just try on this Punnett square. So, now I will answer that question. I can identify the male parent and the female parent because it's labeled. I can identify the alleles that are passed down by each parent and the option. So there's a B option, a big B option, and a little b option. Dun dun. And I can see there's a genotype or a phenotype of purple for heterozygous or homozygous dominant of purple. And I can see that there's a heterozygous option, genotype, with the phenotype of purple again. There's two of those. Dun dun. Two out of 4 is heterozygous purple. And the last one out of 4, is homozygous recessive with a recessive color of white. I can also identify that this is complete dominance because when the big B is present, everything is the dominant color, which is double B's. And so as long as there's a big B in there, everything's purple, which means that there is complete dominance in that big B. So, is there anything else I can list? I don't think so. I think that's about it. If you've got more, great. Next slide. Can you identify the similarities and differences between these two diagrams? And which one is easier for you? So, I just talked about Punnett squares, right? This is a parent. I don't know which parent because it's not labeled. This is also a parent. I don't know which one because it's not labeled. And they're both heterozygous. Big letter, little letter. And this is obviously another color thing because there's this gold color and this white color. There's a homozygous dominant, homozygous recessive, and heterozygous. 2 out of 4 again. So this is the same breeding concept as last time, at least parents-wise. And so you see that here demonstrated on the left-hand side by like this ancestral diagram, which might be easier for you to use rather than the Punnett squares, which is great. That's fine. And you just do that by giving 2 options for each little piece gets 2 options. So this one has 2 arrows, 1, 2. That one has 2 arrows, 1, 2. This one has 2 arrows, 1, 2. That one has 2 arrows, 1, 2. Very similar concept. If it makes sense for your brain, great. If not, also great. Just use the Punnett squares. Now we're going to talk about generations and kind of see what the generations do. So, pause the video, kind of see what you can find. Dun dun dun. Okay. I assumed you paused video. If not, not a big deal. Generation 1. We're crossing homozygous parents. And you see this. This is homozygous dominant. This is homozygous recessive. And then 100% of the offspring are AA, or heterozygous. Cool. A, big A, little a, big A, little a, big A, little a, big A, little a. Generation 2. Now I'm crossing 2 of the first generation's offspring. Because 100% of the offspring is heterozygous, I am breeding 2 heterozygous parents. Which means I get the pattern that I've seen in the last couple slides. 1 homozygous dominant, 2 heterozygous dominant, and 1 heterozygous recessive. Now, this concept is trying to identify which parents, what would happen, or what you would expect to happen if you didn't know the genotype of a dominant parent and you wanted to find out. So how could you deductively reason this? How could you work backwards? The puppies are excited. It's okay. I'll let them be excited. They're being silly gooses. Pause the video. Take a peek. See if you can't understand it. Okay. So basically what they've done is if they know the recessive phenotype is P. P is a bad letter to use for these things because it's really hard to tell the difference between an uppercase P and a lowercase p. But if we know the white color only shows up when there's 2 little p's, which means that it is truly dominant. This is true dominance we're talking about. And we don't know if this male, right? This male, it calls it male. Calls it male. If this male flower is heterozygous or homozygous. If we were to breed these 2 things, to figure that out, what would we expect to get? So if it was 2 big letters, it would be, we would expect to see 100% heterozygous dominant. Right? 2 little p's, 2 big p's means everyone gets a big P and everyone gets a little p. What we would expect to see if this was 2 big p's, no, a big P and a little p, and then a big P and a little p. We would expect to get 50%. So 50% heterozygous, 50% homozygous. I feel like I've misunderstood something. 2 little p's. I see. So these 2 are different. And these 2 are different. Anyway. Now you know. Now, do you understand the differences between these 2 concepts? And can you explain it? Just take a peek. Now that you've paused the video and tried to learn, you see that the C's are consistent all across. And so we're talking about the same gene. And we're, in this case, talking about the same genes with two different letters on them. So this would be like a big letter, and this would be a small letter in the cases that we've been using. And so if you breed for codominance, which makes the colors the same, or makes a new option of color, doesn't mix them together, but it doesn't mix them together in a pattern, but codominance makes a new color altogether, mixes the colors to make a new color. Means that if someone gets a big letter and a little letter, or an R and a B, meaning both standing for two different alleles, you would get a new color throughout everything, if it's heterozygous. Incomplete dominance, as you can see, demonstrates that when you get a letter, or a big letter and a little letter, or an R and a B, you get a pattern of the two colors. You don't get purple, but you get blue and red on the same being. And can you describe what true dominance would look like in this case? Right? True dominance would look like either all blue or all red. All a big letter is basically what that would look like. Because they'd all be heterozygous. Ta-da! Now, we have not talked about genotypic ratio or phenotypic ratio yet, but pause the video, see if you can't guess what they mean. Dun, dun, dun. Yay! You're back. Your guesses. What are said guesses? Drop in the comments, I guess. I don't know. I'm pretty tired today. Let's see. Genotypic ratio. All heterozygous. Phenotypic ratio. All purple. Let's look at this. So parent 1, both parents are homozygous, dominant, or recessive. We've got a parent of each, which means each one is going to get a big F. Each one is going to get a little f, which we can see by the photos. That means they're all purple. So genotypic ratio is what the letters are. So 100% of these flowers are heterozygous. The phenotypic ratio would be 100% of these flowers are purple. Trick question. What kind of dominance is this? Do you know? Dun, dun, dun. What's your guess? Any guesses? Okay. The guess. So, incomplete dominance is when two colors make a pattern. They don't overlap to make a new color, and they don't just omit a color. They make a pattern. And these flowers obviously aren't patterned, so this is not incomplete dominance. So that leaves us with codominance, which is a whole new color option, or true dominance, which means that these flowers are just the color of the dominant parent. And because of that, we need to look at the dominant parent's characteristics. Dominant parent. We have no information on what their characteristics look like. So, we can't say for sure if this is true dominance or codominance because we don't have enough information. given more information, we could figure it out. That being said, we're going to jump to the next one, which is new and interesting and things. If you want to pause now to kind of take a guess and take a gander, you can. But I'm just going to go into explaining this and kind of pause, so you can kind of pause and kind of think ahead and do the things. So, this is a bigger Punnett square than we're used to. And you can see this one. This parent is called a round yellow. It's a Punnett square and a pea plant. And I happen to know that this is talking about the peas in the pea pod, because this is a really common genetics example, because it's easy to manipulate in the lab. And the other parent is a round yellow, but they are heterozygous. And what's that mean? That means heterozygous. But you'll notice that we have round and we have yellow. Those are two different traits. So what we've done is we've paired traits together and made a bigger Punnett square for all the options for both traits. What would it look like when you start incorporating an extra piece, right? So you'll see this a lot in the color examples that I give. So if it's a little convoluted, we'll get more examples. But in this case, everyone still gets each option from a parent. So they need a big R and a little r, but they also need a big Y and a little y. Big Y, little y. And you need to line up enough options for everyone to have, line up enough rows for all the letters. So we've got 4 letters here. We should have 4 rows, because big R, big Y is an option, but little r, big Y is an option, right? And big R, little y is an option, because now we've added double the genetics. And you can Google it. There's practice online. We will do more practice with true color genetics, and we will go slow. I promise. And it's a little hard to see, but you can see the round yellows are labeled. The round greens are labeled. The wrinkled yellows are labeled. And the wrinkled greens are labeled. And this one would be truly like homozygous recessive for both genes. This one is homozygous dominant for both genes. This row here, that goes across this way, is truly heterozygous for both genes. And then these rows here are dominant in one gene, recessive in the other. These are dominant in one gene, recessive in the other. What else? Oh, I can tell by looking at this, because there's no patterns and no mixing of the genes, that this is talking about true dominance. Because the yellow stays yellow, the green stays green, the round stays round, and the wrinkled stays wrinkled, no matter what the gene variation looks like. So both of these things we're talking about true dominance, which makes it a little easier to concept practice. And then, I won't go over this in very, very major detail, but you could take a look at this. This is what happens when you cross, when you start looking at 3 genes at a time. In this case, 3 genes that are heterozygous, in everything. And you'll start to see some patterns form in all of these. The more practice you get, the easier it's going to understand. And this is all statistics. So the more breeding you do, the closer you are going to be to like that 50%, that 20%, that 30% rate. That 9 out of 64 births, that 27 out of 64 births. You know, if you were to do one birth, of course you're not going to get every single option because it's just one. You're going to get one option. And this one I like because it demonstrates the colors across the board. So you can see that you have even if you're not very good with numbers, you can see that there's a lot more gray on this board, which means it's going to be a lot more likely. But you can also see that there's quite a bit of orange and pink and tan, which are all just as common, and so on and so forth. What did we learn? Drop in the comments. We learned, at least what I learned, practice makes advanced genetics easier to understand. Going slow, taking your time, practicing, makes it easier to actually nerd out and figure out why animals like these actually have these fun, cool patterns and things. And how to achieve the fun, cool patterns if you want to do that in your breeding program. And learning color genetics and simple genetics also allows you to then bring in more things like motherly instinct and genetics related to that, and how fast things grow up, and a lot of these things that are important in agriculture genetically, but we don't really talk about because color genetics is funner to learn about when you first start learning. So, yay, we did it. Another video, lesson learned. I hope you learned something. I hope it was helpful, and we will see you next time. Bye!